Colour deficient vision. People with normal colour vision are referred to as trichromats. They are sensitive to all three systems: blue, yellow, red, green and black, white. However, about 7% of the male population and 1% of the female population have a deficiency in the blue-yellow system, the red-green system, or both. This deficiency is caused by an absence of the hue-sensitive photopigment in certain cone types. A dichromat is a person who is colourblind in only one of the systems, blue, yellow or red-green. A monochromat is sensitive only to the black-white system and is totally colourblind. Most colour-deficient people are dichromatics and have their deficiency in the red-green system. Colour blindness tests typically employ sets of coloured dots such as those in figure 5.13, which you've probably seen before. It's like a circle that's filled with dots and then there's a number in a different colour, also made out of dots. A person who is red-green colourblind cannot discern the numbers embedded in the circles. I've never met anyone who is a monochromat and only sees black and white. I didn't even know they existed. Same as I'd love to meet someone who can smell numbers or taste numbers or something like that. Anyway, I'm going off tangent. In the news, that dress, blue and black or gold and white? In 2015, a photo of a dress posted online caused a social media storm. If you haven't seen it, search for white and gold or black and blue dress. Some people saw the dress as blue and black, while others saw it as white and gold. When I show the images in lectures, the class is typically evenly split regarding what colour combination is seen. Also, most people are stable in terms of the colours that they see, although ambient lighting conditions and the type of device they view it on can influence their percept. Is it percept or percept? Because I'd never actually seen the word. I've seen perception and perceive, but never percept, until this textbook. That said, some people can alternate between the two colour combinations. I see white and gold. I've always only ever seen white and gold. I can't see blue and black. What do you see? The basis of this illusion is colour constancy. As previously discussed, the retinal image, which is the direct information that the brain has about objects, can constantly change due to changes in viewing conditions. Since these changes don't indicate actual changes in the objects, the brain has to be able to actively interpret the changing retinal image so that we can perceive the constant, unchanging object, rather than the ever-changing retinal image. For example, it needs to take into account viewing distance to generate a constant percept of size. With colour, the changes in viewing conditions that the brain has to deal with are variations in the spectral composition of the light that illuminates the object. I wonder if that's referring to if we stare at something and the image didn't bounce around on the back of our retina, it would disappear if we looked at it for too long or jump around. Okay, the colour we perceive is based upon the relative combinations of wavelengths that an object reflects. For example, we perceive something as blue because it reflects more light at the short wavelength end of the spectrum than at the long end, which is red. However, the amount of short wavelength light that the object reflects also depends upon how much of that wavelength there is in the light falling on the object, i.e. on the spectral composition of the illuminant. The illuminant is the light shining of the object. For example, while most light bulbs emit light over the entire visible spectrum, the different types emit most of their light within certain parts of the spectrum. A white LED light emits most light at the short wavelengths, and a cool white fluorescent light emits most light at the long wavelengths. Similarly, the spectral composition of sunlight changes during the course of the day, with the short wavelengths being progressively lost as the sun drops toward the horizon, which is the reason why sunsets are dominated by the long wavelengths and look so red and yellow. However, we don't see the colour of objects change due to changes in the illuminant. An object doesn't look bluer when it is being illuminated by an LED rather than a fluorescent light source, even though more blue light is being reflected off it. Rather, the brain can take into account the changes in the spectral composition of the illuminant, as long as the light still contains all wavelengths, by comparing the different wavelengths reflected by all of the objects in the visual scene. So colour constancy can account for why our percept doesn't always correspond to the actual retinal image, i.e. the actual wavelengths being reflected by the object. But the really novel and interesting aspect of the dress illusion is, of course, that people see different colour combinations. We see the world differently in a fundamental way. This seems confronting to a lot of people. There are a number of visual illusions in this chapter, but typically everyone gets the same illusion or can alternate between two possible percepts. Example, the bistable percept in figure 5.30. But with this dress illusion, different people are actually seeing the world differently, even though it appears to be a fairly straightforward image. While there has been active debate regarding the basis of the illusion, it appears to be due to differences in the assumptions made by the visual system in applying colour constancy. That is, different people are making different assumptions about the spectral distribution of the illuminant. Recall that these assumptions occur at the subconscious level. Analysis and reconstruction of visual images. Once the transformation of light energy to nerve impulses occurs, the process of combining the message received from the photoreceptors into the perception of a visual scene begins. As you read this page, nerve impulses from many neurons are being analysed and the visual image that you perceive is being constructed. How does all of this occur? From the retina, the optic nerve sends impulses to the cortex via the thalamus, mainly to the primary visual cortex, area V1, located in the occipital lobe at the back of the brain. Retinotopic mapping occurs in that neighbouring cells in the retina project to neighbouring cells in V1. Given the topographic mapping in the retina, that is, neighbouring cells in the retina are looking at neighbouring regions in the visual world, this retinotopic mapping thus leads to topographic mapping in V1. This mapping is not uniform, with the fovea taking up large area of the cortex, consistent with the high density of cones and retinal ganglion cells in the fovea. Neurons in V1 are tuned to particular aspects of the visual scene, with orientation information being particularly important in V1 neurons. They respond optimally to a particular orientation and to a range around it, which defines the neuron's tuning bandwidth. Daniel Hubel and Torsten Wiesel were the first to discover this tuning property of V1 neurons, and also that they are arranged systematically. Neurons that have their receptor fields in the same location and have the same preferred orientation tuning are arranged in columns, with neighbouring columns having a slight difference in their preferred orientation. Hence, a collection of these columns, called a hypercolumn, contains the neural machinery to encode all possible orientations within a given region of space. In 1981, Hubel and Wiesel were awarded the Nobel Prize for these findings. V1 neurons have very small receptive fields, with the largest ones being around 2 degrees of visual angle, which is about the size of your thumb at arm's length. The perception of any object thus involves the integration of the response of many neurons, each of which is analysing a small region of that object. This process is made easier given the topographic mapping in V1. These pooling processes occur in areas outside of V1, which results both in the neurons having larger receptive fields and a progressive loss in topographic mapping at higher cortical levels. It was initially thought that each neuron in V1 detected a particular feature, such as orientation, and other neurons combined the output of these neurons to become sensitive to more complex features, culminating in neurons that are sensitive to specific, complex objects such as your grandmother. While an attractive concept, it is wrong. V1 neurons are not tuned to a single feature, but to a various combination of them, including stimulus width, length, motion detection, and speed. Different combinations of these features will elicit the same response from a particular neuron, meaning that the output of a single neuron cannot indicate anything about a particular feature. Also, if being able to perceive your grandmother depended upon a particular grandmother neuron, you would need a dedicated neuron for every complex object. While there are a huge number of neurons in the brain, given the effectively infinite number of objects you can perceive, you would quickly run out of neurons. And how would you then perceive new objects? Our ability to perceive objects actually depends upon the activity of a number of neurons, that is, a population or response network. This is true even for simple features, such as orientation. If orientation perception was based upon the activity of a single neuron, our resolution would be limited to the difference in the preferred orientation turning of neighbouring columns in a hypercolumn, which is about 15 degrees. That is, they like saying that over and over again, that is, the minimum difference in orientation that we could detect would be about 15 degrees, which would make reading an analogue clock different given that when the minute hand moves by a minute, it changes its orientation by only 6 degrees. Instead, the visual system uses a population response. The tuning bandwidth of orientation selective neurons means that a number of orientation-tuned columns will respond to a particular orientation, and so the brain can use the average of that population response to perceive orientation at a finer resolution. The tilt after effect demonstrates the population encoding or orientation. Figure 5.14. This figure has 4 boxes. It's on page 159, and the first box is diagonal lines leaning to the left, the next box is vertical lines, the next box is vertical lines leaning to the right, and then the last box is vertical lines again. And it's called the tilt after effect. Different neurons are sensitive to different aspects of the visual world. Examples, some are sensitive to colour and others to motion, and they provide input to specialist parallel pathways. See Australian focus. Thus, as a green and gold beach ball sails towards you, separate but interacting modules in the brain are simultaneously extracting its colour, shape, distance and movement, eventually constructing a unified percept of its properties. This information is ultimately projected to higher levels in the brain, specifically the visual associate cortex, where the stored knowledge is used to interpret the visual percept. If all goes correctly, a process that began with nerve impulses from cones now ends with our recognising the beach ball for what it is and catching it. What a process to go through that we just take for granted that happens so sort of simultaneously, instantaneously. And the last part of this section on vision is the concept check. So it just goes over those key terms. Our light-sensitive receptors are located at the back of the eye in the retina. There are 2 types of receptors, rods and cones. Rods have the greatest sensitivity to light, so are active at night, while cones are active in daylight. Rods do not result in the percept of colour, while cones are most numerous in the fovea and give us our ability to see colour and fine spatial detail. Our ability to see a huge range of light levels is further aided by light and dark adaptation. The decrease and increase respectively in the sensitivity of the rods and cones is due to increases and decreases in light levels. Humans have trichromatic colour vision in that our perception of colour is based upon the relative activation of our 3 cone types. At the cortical level, this relative activation is encoded via 3 opponent mechanisms: red-green, blue-yellow and light-dark. Visual neurons are specialised to detect specific aspects of the visual scene, but each neuron encodes multiple features. Our ability to see things is always dependent upon the response of a number of neurons, that is, a population response.